Hello friends, welcome to Easy Nurse, where nursing education made easy. So in this session, I'll be discussing the most important question of microbiology in post-basic BSc. So as we know, in the post-basic BSc, microbiology is kind of a little tough subject, it presumes, but I'll be telling you guys such points and some easy questions and the important question which if will study, then obviously you will get 100% attempt in question. Okay, as well as I will tell you guys what are the tips and tricks to remember the microbiology questions. Okay, because it's kind of little hard to remember more other subjects. Okay, and also in this session I'll be telling you guys different kind of tips and tricks, okay, to remember the microbiology important questions and answer, because other more than other subject, microbiology seems like kind of a different hard subject. Okay, but don't worry, I am here to make you guys study. Okay, so just let get jump into this session. So we'll be starting with our microbiology different kind of chapters. Now if we look into the microbiology, there'll be different type of important chapters, especially immunity chapters. Okay, then it will come staining chapters, okay, how to identify the microorganisms. Then other chapters like diagnostic examination, as well as different kind of microorganisms. So as we know, microorganisms are divided into 5 types. Okay, so that is the basic knowledge everyone will have. But what are the important topics? Okay, so as we will start it with the immunity. So as we know, immunity is a defense mechanism of our body, okay, which will fight against the bacteria and different kind of pathogenic microorganisms such as fungus, virus, okay, algae and different kind of amoeba, this kind of things, okay, and protect our body from the harmful pathogens. Okay, so in our immunity, what are the types of immunity? We will just say it first. So the definition says immunity refers to the resistance exhibited by the host towards the injury caused by microorganisms and their product. So if we understand the definition, we will able to know. So immunity is kind of a mechanism in persons or host body, okay, which is exhibit the host towards the injury which will be caused by different kind of microorganisms and their product like toxic substances. Okay, so immunity have different types. So first we are dividing immunity in 3 types, that is innate immunity, adaptive or acquired immunity, and herd immunity. Now innate immunity, it will be from the birth itself, okay, and acquired or adaptive immunity, it can become from by like it can be because of different kind of vaccines. Okay, it can be come from like after one experience of in disease, you may develop different kind of antibodies inside you that will be causing the development of acquired immunity in your body. Then we have is herd immunity, which is actually not kind of a immunity as such, but if you want to understand, like if I am surrounded by some people who are affected, then I will also get affected. But if I am around someone who all are not affected, okay, and they are avoiding the affected people, then so the immunity which will build up on the side of mine or of the environment of mine, that we will call as herd immunity. We will be discuss it later. So in the innate immunity, what are the things? As I told, you can see herd immunity here. So these green persons are actually like fortunate person or the person who have the herd immunity. Now because of this blue person have the acquired immunity or maybe the vaccination. So this red people have infected virus or any microorganism. So what happening now, because of this people will not able to transfer the microorganism into the blue peoples, and the blue people is like the green peoples are surrounded by the blue people. So blue people will not be transferring any kind of, or there is no chance that the microorganism will be transferred to the green person. So anyhow the green persons are being not affected. Okay, so that is kind of a herd immunity. The next one is immunity. So in the innate immunity, again as I told, it is from the birth. So there is also 2 types are present, that is specific and non-specific. In the specific immunity, there will be different types of specific innate immunity. First one is intact skin. Because of our intact skin, so no microorganism will able to directly being in contact with our inner body. Okay, so that also work as a protect barrier. Then we have the ciliary clearance, like our lungs. Okay, there will be always mucus will be present, by which the pathogenic microorganism and toxin will be cleared, or will be through our cough will be expelling it, right? So by that the microorganism will be removing. Okay, so that are ciliary clearance. Then low stomach pH. Okay, sometimes our low stomach pH, what it will like, if the stomach pH is very low, that means the stomach pH is acidic, so it will also help to kill some of the microorganisms. Then lastly we have lysosomes. Now lysosomes will help to engulf the microorganisms. So these are the specific type of innate immunity. Now if we move to the non-specific immunity, there are different kind of white blood cells. Okay, like as monocytes. Okay, then lymphocytes. Okay, these are the different kind of white blood cells are present in innate immunity. Now come to the adaptive and acquired immunity. So now it have again two types, that are active and passive immunity. Now in active immunity, again two types are there, that is natural and vaccination. Now how can I explain it in the sense, any kind of fever if you have go through, so like that same thing, okay, it would be not there in the second time. Like for the smallpox or chicken pox, as you say, if someone had already faced chicken pox, then the second time he will definitely not get it. Okay, or if someone have vaccinated of the vaccine, get vaccinated through the chicken pox or smallpox, he will not get it. Okay, so that is the difference between natural and vaccinated active immunity. Okay, then we have as passive immunity. So now passive adaptive or acquired immunity have two types, that is maternal and one is immunoglobulin treatment. Now in the passive one, okay, what's happening from the in the maternal one, okay, what's happening from the mother, the child is getting the antibodies. Okay, but in the immunoglobulin treatment, because the person can't get it from the mother, what we are doing is we are like from passively or through injection or different kind of antibody treatment, we are giving the immunoglobulins treatment to the person to increase the antibody. So these are the different kinds of immunity. Now we will move on to the acquired immunity, which is very important. So acquired immunity, which is also known as adaptive immunity, is a component on the immune system, okay, that develops different types of the exposure to specific pathogens or the foreign substances. Unlike the innate immunity, which provides a general defense, acquired immunity is highly specific and has a memory. Now see, to understand in innate immunity what was happening, it is from the birth, okay, and it can't resist all of the microorganisms, but because acquired immunity has some memories in it. Okay, so because of that memory, the acquired immunity will able to understand or will able to recognize the particular type of pathogen that have already entered in the body, and the antibody he have already prepared. So they will fight against the microorganism. Okay, so that is little tough and little different, and it is very strong than the innate immunity, and it is a very quicker and more effective response if the same pathogen is encountered again in the future. Okay, and there are two main types of acquired immunity, as I told, active and passive. So now what are the mean of active immunity and passive immunity, we will know in detail. So the active immunity, it will occur when the immune system is directly exposed to an antigen, such as through infection or vaccination. Okay, so as I told, in active immunity what we are doing, we are injecting through it through the vaccination, okay, or maybe person have already experienced. So through the vaccination and all, we are already exposing our body to that particular pathogen. Next, the body will create a specific kind of antibodies and the immune cells like T cells, okay, to fight the infection, and a memory of the pathogen is formed. Then what will happen? This can be natural form of exposure to the pathogen in the daily life or an artificial form of the vaccination. Okay, so this is about the active immunity. Next we will move on to the passive immunity, in which this involves the transfer of antibodies from the one person to another person. Since the immunity does not actively produce its own antibodies, the passive immunity will offer the temporary protection. Okay, and so the examples which include the maternal antibodies passed to a baby through the placenta like breast milk and immunoglobulin treatment and certain kind of disease. Like I told you, like in mothers and all, they will providing the partial immunity. Okay, so this is kind of a passive immunity. So this is all about the acquired immunity. The next important question is sterilization. So to understand the microbiology, we have to understand about microorganism as well as how we have detect them and how we can kill them. Now to kill them, there is different and various types of microorganism killing process, okay, which is also called as sterilization, okay, which is mean the removing or deleting all the microorganisms from a place or from a surface. Okay, so whether they are being in a vegetative form or in spore form, okay, whatever they are. So the definition says it is the process of removing or killing all forms of microbial life, which is including bacteria, viruses, spores and fungi in the form of a surface, which is liquid, medication or biological culture media. So this process ensures that no viable microorganisms are present. Thus, it will prevent the contamination and infection. Okay. So if we are sterilizing a place, we should be sterilized properly, so no forms of microorganism will be there, and there will be a 0% chance, okay, 10 to 0% chance, so that any kind of infection will not be there or any kind of contamination will not be happen. Okay, so that is the definition of sterilization. Next we will move on to different kinds of method. Okay, so there are physical method, chemical method and combined method. Physical method are using by moisture, then heat, pressure and different kinds of thing. And in the chemical, we will be directly using the sterilizing like alcohol, okay, rubbing alcohol and all, or ethanol, or different kinds of other types of acids and all. This all things we can use as a sterilizer. Okay, now in this one, the dry heat sterilization is very important, so we will learn about it. So the dry heat sterilization is a method where we are utilizing high temperature. It extends the period of destroy the microorganisms. So in a very high temperature, okay, dry heat. Remember that no moisture or no vapor we are using, no kind of liquid. Okay, it is completely dry heat we are using. Okay, up to a certain period of time, so that all the microorganism will be killed in that time. Okay. Unlike the moist heat sterilization, dry heat does not involve water or steam, as I told, and it is particularly suitable for materials that can withstand high temperature without the degradation, such as glassware or metal instruments. Okay, so this is all about the dry heat sterilization. Now, in the dry heat sterilization, the most important one is hot air oven. So what is that? Hot air oven is a machine where the direct heat, okay, direct air heat will be used. So what is the principle of this? It uses dry heat to denumerate the proteins, causes oxidative damage and cellular components, such as effectively killing the microorganism. Okay, so what is the procedure we are using? Okay, so the principle is to kill the oxidative damage or cellular components, okay, or different kind of proteins. Now in the procedure we are using, we are placing in an oven which is exposed at a temperature of 160 degree to 180 degrees Celsius just for 1 to 2 hours, and all the microorganisms will be killed or the organic matters will be damaged. Okay, so this is about the hot air oven. The next method of dry heat we are using will be incineration. Now incineration, especially in Hindu people, you may have seen, okay, they will be incinerate, or they will be fully burn the body, okay, dead bodies and all. That's kind of a incineration. or the waste products that we are directly burning, okay, with the help of maybe some chemicals and all, that will be called as incineration. So what is the principle of this? It will be direct exposure to flames, which is in a very high temperature and completely oxidize the microorganism to convert them into the ash. Okay. Then the procedure includes it infected waste, contaminated materials and disposable are burned at the highest temperature in up to 1000 degrees. Okay. And next we have is glass bead sterilization. Now what is glass bead sterilization? Okay, so this is a new kind of a thing. Okay, you may have heard, so listen it properly. Okay, so the principle says the small glass beads are heated around 250 degrees Celsius. Okay, so to make you understand, you may have seen some people will not use oil. Okay, they will the fryams you have seen maybe, okay, or papad, they will fry that particular things using the uh, sand, okay, heated sand or heated salt. Okay, these are the kind of a thing. Okay, that's kind of a similar to this. So what they will do, the glass will, they will make it or they will heat around it in 250 degrees Celsius, and it will be transferring the heat into the object placed within them to sterilize. Okay, so if you see one jar will be there. Okay, just for an example, you suppose one jar will be there, there will be glass bead all over there, and inside them there will be put it, which are the stencils you want to sterilize, you will be putting in them. Okay, and after that what you will do is you will start heat the glass beads. Okay, at the temperature of 250 degrees Celsius, and from the glass bead it will be, the temperature will be transferring to the instruments. Okay, so that is the principle. Now the procedure, the instruments are placed into the beads for a few seconds, okay, to achieve the sterilization. It doesn't need much more time because it is already like the temperature is very high. Okay, and the last one is flaming. And in the flaming, the principle is it is a brief, okay, exposure and exclusive exposure to an open flame to destroy the surface of microorganism, and the procedure includes the instrument like insulating loops and pass through a flame until red hot to kill any adherent microorganism. So in such cases, like if it is something is not sterile, so what we can do is, so if it is any kind of instrument, okay, which is made out of metal, we can directly, or metal or anything, okay, which is heat durable, that we can directly put it into the flame, and until and unless it's becoming red hot, we will not remove it from the flame. Once it became red hot, it will be sterilized. Okay, it will be fully sterilized. So that is the flaming method. Okay, so this is all about the different kind of sterilization method. Okay, next this question is it came 5 years. Okay, last 5 years it have been came, okay, 2 to 3 times. Okay, so the detailed overview of hepatitis B virus. Okay, as you know, hepatitis B virus will be actually affecting the liver. Okay, so to understand that hepatitis B virus, we have to understand the morphology. So the structure and composition of hepatitis B virus is a double stranded DNA. Okay, it is a double stranded DNA virus which will be, so hepatitis B is a double stranded DNA virus which will be belong to the Hepanaviride family. Okay, and it has a outer envelope which have HBsAg or hepatitis B surface antigen and a nucleocapsid code that contains the viral DNA and enzymes and necessary for replication. Okay, and this envelope protects the virus from the outer damage. Okay, so this virus will be transferred through different kind of mode, okay, such as blood to blood contact, okay, which is a direct contact, then sexual transmission, okay, vertical transmission, and different kind of other modes. Okay, so other modes as such through the needles and all. Okay, so how this is spreading, okay, that we will know. So to understand that, we have to know the pathophysiology of this virus. Okay, so after entering the bloodstream, HBV will target the liver cell, as I told, okay, which is hepatocytes. Okay, then it will integrates the DNA into the liver cells, okay, and it will integrate, like it will be duplicate or it will be entering in the DNAs of the liver cells, okay, like genomes replicates and releases more virus and infect the other cells of the liver. So, the immune response to HPV cause a liver cell damage leading to inflammation of the liver cells and the damage, a fibrosis or chronic cases such as cirrhosis and liver cancer. So, this is the main pathophysiology of the hepatitis B virus infection. Okay. Next, we will move on to the clinical manifestation or signs and symptoms. So, in the acute infection or the first stages of the infection, the symptoms can range from the mild to severe, include fever, fatigue and dark urine as well as the abdominal pain. And in the chronic infection stage, there will be many cases are asymptomatic initially, but long-term infection can lead to cirrhosis of liver, liver failure and hepatocellular carcinoma or cancer. Okay. Then in the asymptomatic carriers, which will some individuals may carry the virus without showing the symptoms but can still transmit it to others. Okay. So this will be the clinical manifestation of hepatitis B virus. Now how to diagnose this? So to diagnose this, there will be two processes, that PCR testing and serological test. So PCR testing include HBs antigen, okay, then anti-HB antigen, then the HBe antigen and antibody to core antigen. So these are the kind of serological tests that will be done for the hepatitis B vaccine, hepatitis B virus infection. Okay, then treatment. Okay, in treatment, it will for the acute hepatitis B, we can often does not require any treatment and most healthy adults recover by their own. Okay, sometimes it will be recovered by their own, that much treatment as per not needed in the acute cases, but in the chronic cases treatment includes antiviral medications such like as tenofovir and enectovir, which can reduce the viral load and prevent the liver damage as well. Okay, and in the other than that, we can go for monitoring like regular liver function test and screening for the liver cancer are essential for chronic HBV patients. Okay, so other than the treatment, we can prevent it before it occurs by the vaccination, screening, safe practices. Okay, so these are the things we can do for a HBV patient. Next is the description of the bacterial cell wall. As I told you, you have to know more about the microorganism as well. Bacteria is the most important, important kind of microorganisms. Okay, so to know that, we have to know about the bacterial cell wall, and this question have came at least 4 to 5 times in the last 10 years. Okay, so remember what I am telling. Okay, and if you are missing something, get again to the video, okay, revise it properly. So the bacterial cell wall is a rigid protective and structure that provide the shape, support and protection to the bacterial cell. It is essential for the survival of the bacteria, particularly maintaining the cellular integrity against the environment stress. As we know, for the every cell, the bacteria in the bacteria as well also, like the cell wall is protecting the shape as well as they're protecting it and giving different kind of supports. Okay, so the structure or the composition of bacterial cell walls are vary between main two types. Okay, so now bacterial cell wall are different types. Okay, based on the which type of bacteria it is. So there are two types, that will be gram positive and gram negative. Okay, so the composition, the main component of the bacterial cell wall will be peptidoglycan. Try to remember it. Peptidoglycan will be the main component of the bacterial cell wall, also called as murine. Okay, it is a unique polymer consisting of sugar and amino acids. Sugar in the sense carbohydrate. Okay, carbohydrate and amino acids. Then in the peptidoglycan layer, this consists different kind of chains. Okay, it consists of two types of alternating sugar chains and N-acetoglycosamine or NAG, and also N-acetomuramic acid, like that together with people the peptide chains that form a mesh like of structure which will be providing the rigidity and to the cell. Okay, So as I told, there are two types of cell wall. In the gram positive, the peptidoglycan layer will be very thick. There will be teichoic and liptoic acids, and it will stain in the gram staining. It will be staining in purple. And for the gram negative cell wall, the thin peptidoglycan layer will be present. Okay, outer membrane will be there, and it will be stained as a pink or red. Okay, so these are the different types of bacterial cell wall. Now, what is the function of the bacterial cell wall? As we know, the structural support it will provide. So, it will prevent the cell from lysing or bursting or in the hypnotic environment by maintaining the cell shape and the rigidity. Okay, so it will provide support and the structure in the different kind of environment. So, the cell damage will not be there. Okay, or the cell damage will not happen. Then protection, the shield will save from the physical damage and contributes to the defense mechanism against the host immune system. Okay. So, if some bacteria is entering inside the human body, it's protecting the bacteria from the defense mechanism of the host. Okay. And it is also work as a selective barrier. Although it is permeable, some substances, the cell wall can be regulated as a passage of larger molecule as well. So, these are the main functions of a bacterial cell wall. So, with this important question, I am ending this session, but do not forget to attend live class of microbiology as well. Here I will be discussing more such kind of content, and I will be discussing the most important one as well. Okay, I will be repeating whatever the doubts you want to clear, I will be clearing there. Okay, so stay tuned with Easy Nerds. Keep learning and keep exploring with us. Thank you for this session.